Hello everyone. We are going to start a new course, Plant Bacteriology, and this will be our first lecture. In this course, we are going to learn all about an important pathogen that is bacteria. So, to study this course well, you should refer certain books. I have listed a few books for this course. Of course, the list is not exhaustive here. There are many books which can be good. You can refer to any other books where the content is good. So you can go through the other books as well. In fact, there are many other books on plant bacteria these days. I have just given you some names of books that you may refer. Now, to know the subject, we always go to the historical perspective of the subject. The history of plant bacteriology can be traced back to the time of Anthony van Leeuwenhoek, when he first saw bacteria in the dental plaque in the year 1676. He was basically a cloth merchant from Holland. His hobby was to make lenses. With his own simple microscope, he could see bacteria and also protozoa. He termed them as little animalcules, and he made drawings of all the living microorganisms that he saw with his simple microscope. He is known as the father of microbiology because he is the discoverer of the microbial world. So he is the discoverer of bacteria, he is the discoverer of microbial world. Before him, there was no idea about microbes. So it was Antony van Leeuwenhoek who is the pioneer, who is the father figure of microbiology. Though his work is not related to plant bacteria as such, but this is the beginning to search for bacterial pathogens. Robert Koch is another very important name in the field of microbiology. Uh, he was actually a German bacteriologist. Again, his work is not related to plant bacteria, but his observations and his discoveries led way to other field of sciences, like he established Koch's postulates, through which we now, even now, implicate a microorganism to be a plant pathogenic or animal pathogenic ones. To know more about Koch's postulates and the modifications, the molecular Koch's postulates, you can refer to one of my YouTube videos in my channel. The details have been discussed there. Robert Koch, he discovered bacteria causing anthrax, cholera, and tuberculosis. So these are all important diseases at that time. And he discovered the fact that the bacteria cause all these diseases. He used gelatin as solidifying agent. These days we use agar-agar to solidify our media, but it was gelatin which was used first, and it was Robert Koch who started using gelatin. He is called as father of bacteriological techniques, and he was awarded Nobel Prize in the year 1905. So the contribution of Robert Koch has actually opened the field of microbial studies in large scale, and many of his students also contributed a lot in the development of the science microbiology as a whole. Louis Pasteur is again another microbiologist from France. He pioneered and founded modern bacteriology. He disproved spontaneous generation theory and established germ theory of disease. This germ theory of disease says that there will be one microbe associated with a disease. That is the germ theory of disease. Without any microbe, disease cannot be caused. There is a germ behind every disease. That was the observation. He gave pasteurization technique. Pasteurization is basically a killing of microbes, though this technique does not kill all the microbes present in the substance that you are targeting to, but this technique is used to minimize the load of microbe from food substances like milk. He developed vaccine against anthrax and rabies. This is one very important achievement of Louis Pasteur. He also discovered the role of microbes in fermentation. He is the father of modern microbiology, Louis Pasteur. Then we are all aware of this gentleman, Alexander Fleming. He discovered antibiotic penicillin. Penicillin, that is the wonder drug, in the year 1928 from one fungus, Penicillium notatum. Now, this penicillin, an antibiotic, it kills bacteria, especially gram-positive bacteria. This is the first antibiotic discovered ever, and it saved thousands of lives during World War II. He also discovered lysozyme, one enzyme. This enzyme also can kill bacteria. He was awarded Nobel Prize in the year 1945. He was a Scottish bacteriologist. So now we know that bacteria were discovered by Leeuwenhoek in the year 1676. MLO or Mycoplasma like organisms were discovered by Doi et al. In the year 1967, almost 300 years after bacteria were discovered. Spiroplasma is again another prokaryotic microorganism. It was discovered by Robert E. Davis in the year 1976. There are, in fact, hundreds of contributors to the science of microbiology in general and also in plant bacteriology. Some important discoveries or inventions have been mentioned here in the form of a table. Tyndallization. We know that tyndallization is a technique of killing microorganisms to sterilize substances like soil. If you want to sterilize some soil, then you have to follow tyndallization technique. It's a repeated heating and cooling technique. Tyndallization is a method of sterilization. So this technique was devised by John Tyndall. Now all the methods of sterilization in plant pathology have been again discussed in another YouTube video in my channel. You can go through that video to know more about methods of sterilization. Then chemotherapy was discovered. Then Walker, he first tried to prove that bacteria cause plant disease by following Koch's postulates. It was the first attempt to prove bacteria as plant pathogen. Then agar-agar was discovered as a solidifying agent. There were some problems with gelatin that was initially used by Robert Koch. The melting temperature is less and solidification is also not proper with gelatin. Whereas agar-agar is better as a solidifying agent at room temperature 25-30 degrees Celsius. Agar-agar keeps the medium solid. Melting temperature of agar-agar is 44 or 45 degrees Celsius. So the discovery of agar-agar as a solidifying agent in the year 1881 has been a significant achievement in the microbiological lab because it helped the scientists, laboratory researchers to cultivate microorganisms. Then in the year 1878, T.J. Baril he first showed that bacteria can cause plant disease. He was actually studying the fire blight disease of apple and pear. And the bacterium that we know today is Arwenia amylovora. So it was the first bacterial disease to be discovered in the year 1878. Then Petri in the year 1884 invented Petri dish. We all know how Petri dish looks like. And this Petri dish helped the microbiologists to grow and preserve, to isolate, maintain microorganisms very easily. And one landmark achievement in the characterization of bacteria was this gram staining. It was devised in the year 1884. This gram staining gives two types of reactions. One is gram positive and gram negative. So we'll discuss later more about this gram staining technique in the practical. Then the genera were discovered like Pseudomonas, Corynebacterium. Then one important figure in plant bacteriology is E.F. Smith. He validated bacteria as plant pathogens in the controversy with A. Fisher. E.F. Smith is the founder of phytobacteriology as a science. He has a lot of contributions in the subject plant bacteriology. So we can call him as the father of plant bacteriology. Then crown gall disease of plant was discovered in the year 1910. This crown gall disease is comparable to the cancer disease of animals where tumors are formed. The genus Arwenia was established, and then in the year 1928 Griffith discovered the transformation phenomena in bacteria. This transformation phenomenon in bacteria had a long-term effect. In fact, this observation of transformation in bacteria had gone a long way to the discovery of DNA as a genetic material. Then electron microscopes were developed in the year 1930s. Then Xanthomonas as a genus was established. Agrobacterium as a genus was established. DNA as a transforming principle was established in the year 1944. Then Alexander Fleming, who discovered penicillin, was awarded Nobel Prize. Penicillin was the first antibiotic to be discovered. Another important phenomenon in bacterium is conjugation. It was discovered in the year 1946. Then came one Indian scientist, 1948, M.K. Patel, who established a school of bacteriology at Pune, Maharashtra, India, and discovered first new species of a plant pathogenic bacterium, Xanthomonas, on Ipomia. He also reported more than 30 bacterial diseases from India. M.K. Patel can be considered as the father of plant bacteriology in India. Then the plasmid, which is an extra chromosomal covalently closed circular self-replicating DNA. The term was coined in the year 1952 by Lederberg. We know that gram staining is a method of characterizing bacteria to two groups, gram positive and gram negative. But it is a bit long process. Saslow in 1952 gave a quick and confirmatory test for gram staining using 3% potassium hydroxide. It is a quick method and also confirmatory method of gram staining. Then came another antibiotic, streptomycin. Waxman in the year 1952 discovered another antibiotic, and he got the antibiotic from Streptomyces griseus, one bacterium. So one antibiotic from a soil inhabiting bacterium, Streptomyces griseus, was obtained, and we still use this antibiotic streptomycin. In the later part of this course, we will see that there are many antibiotics which are obtained from this bacterium Streptomyces. We will also have a discussion on the sources and the mechanisms of actions of some antibiotics. Another important phenomenon in bacterium is transduction. This transduction was discovered in 1952. Then Delovivrio. Delovivrio is one parasitic bacterium. It is parasitic to some gram-negative bacteria like plant pathogenic ones. This parasitic bacterium was discovered in the year 1962 by H. Stoll. Hypersensitive phenomenon was discovered in 1964 by Clement et al. As we said earlier, mycoplasma-like organisms, which are now known as phytoplasma, were discovered in 1967 by Doi et al. Bacteriophages were studied by many researchers, Luria, Delbruck, Hershey and Chase. They also did some experiments with bacteriophages. Then first patented living organism, Pseudomonas putida. It was done by Ananda Mohan Chakravarthy. This organism, bacterium, was genetically engineered to clear oil spills. It was the first patented living microorganism. In fact, we can say this is the first patented living organism. Then the crown gall disease caused by Agrobacterium tumefaciens was managed by using another bacterium, Agrobacterium radiobacter K84 strain. So it was a biocontrol of a plant bacterial disease. It was demonstrated in the year 1972. How this agrobacterium causes gall formation or tumor formation in plant? It is all because of a plasmid, Ti plasmid, and this Ti plasmid was demonstrated in the year 1974. Then Pathover system of naming plant pathogenic bacteria was given by Dye et al. In the year 1980. Then with the advancement of other branches of science, biological science, sequencing of organisms started, and the first sequence, complete genome sequence of a plant pathogenic bacterium was done by Simpson et al. In the year 2000. It was Xylella fastidiosa, one xylem limited fastidious bacterium. Then sequencing started with other bacterial pathogens like Agrobacterium tumefaciens, Ralstonia solanacearum, Pseudomonas syringae, and so many. And in fact, now out of about 150 plant bacterial pathogens, more than 120 and 30 bacteria have been sequenced. So the genome sequencing has been really very fast. These days we can have genome sequencing done very fast. Within a day, one microorganism's genome can be fully sequenced. Now, let us see how plant bacteriology started in India. It all started in the year 1908 with the Tundu disease of wheat. Then Patwardhan in 1928 reported black rot of cabbage, which is a bacterial disease. Then we talked about M.K. Patel already. He is the father of plant bacteriology in India, we can say. Then M.K. Hingorani in the year 1950 reported complex nature of Tundu disease. Tundu disease is a complex disease. It is caused by a bacterium Clavibacter tritici. It was earlier named as Corynebacterium tritici and a nematode Anguina tritici. These two pathogens can separately cause diseases in plant with their separate identities, with their separate names of diseases. But when they come together, they bring about another set of symptoms and which we call as Tundu disease. Many bacterial diseases of potato, onion, and pea were also studied. Then the Indian report of bacterial blight of rice disease came in the year 1959 from Maharashtra. Vide and Rangaswamy also did pioneering works on plant bacteriology in India. G. Rangaswamy, he authored books on plant bacterial diseases. D. N. Srivastava, 1962, he studied survival and epidemiology of bacterial blight of rice. B.P. Chakravarthy developed specific media for many bacterial plant pathogens. He studied the nutritional aspect of plant pathogenic bacteria. Then J.P. Varma in the year 1970, and for many years he studied bacterial blight disease of cotton. His one significant work was the book called The Bacteria, which was published in the year 1987. This book is rich with lots of information on bacteria, basic informations, and also the applied aspects of bacteria in general and also plant pathogenic bacteria. So, this book is quite important, and I also suggested you to read the book in the list of books to be referred. Now, what is the importance of plant pathogenic bacteria? Should we study and why we should study? Plant pathogenic bacteria cause severe economically damaging diseases like spots, pustules, smelly tubers or soft rotting and plant death even like wilting. There are other diseases which are regulated or caused by bacteria through hormonal disturbances in the plant parts like leaves, shoots, and one prominent example is crown gall, which is simply one hormonal disturbance in the plant body. There are many serious diseases of plants throughout the world caused by bacteria. But as far as number is concerned, the number of plant bacterial diseases are lesser or fewer than fungi or viral diseases. And relatively, plant bacteria cause less damages than fungal or viral diseases. But bacterial diseases gain more importance because we do not have enough options to control them. One very important aspect of management of plant bacterial disease is use of chemicals, but we have limited number of chemicals. Though some copper fungicides can work well for plant bacteria, we have very limited number of them, and many a times those copper compounds cannot be used to to several crops. And these antibiotics, which are effective in the lab, which are effective in vitro, we cannot use antibiotics in the large scale to control bacterial diseases in plants. These antibiotics can cause evolution of antibiotic resistance and the transmission of this antibiotic resistance to human pathogenic bacteria. So, for the fear of antibiotic resistance development, the use of antibiotics on large scale is restricted in agriculture. So, that is the reason why bacterial diseases in plants gain importance. Now, let us see the bacteria and their losses in terms of US dollars. Agrobacterium tumefaciens, annually the loss is 23 million US dollars. Then we have a good number of other bacterial pathogens like Clavibacter, Arwenia, we have Phytoplasma as well, Pseudomonas, Spiroplasma, Xanthomonas. All of them cause heavy losses to the plant because we do not have enough management options. So, this is the end of our first lecture. We will see you again in the second lecture of this course. Meantime, if you have any query, you can write to me in the comment box. Thank you very much.